Chapter 6 is all about cellular respiration. This is the process by which food is used to acquire energy by cells. So the overall equation is shown here. Glucose and oxygen are the reactants, and the products are CO2, carbon dioxide, water, and energy is released. And it's usually stored in the form of ATP. So what exactly is going on here? So what's happening is that the food molecules are being oxidized. So that means that electrons are being extracted from the food molecules. And these are called redox reactions. So the loss of electrons from one substance is called oxidation, and the gain of those same electrons by another substance is called reduction. So the food is being oxidized and the electrons are added to a molecule called NAD+. And that process is catalyzed by an enzyme called dehydrogenase. What's actually happening there is that two hydrogen atoms are being extracted from the food along with their electrons and added to the NAD+. So the two hydrogen atoms have two electrons. One of those electrons neutralizes the positive charge on the NAD+. The other one is just added as a hydrogen atom. And what's left over is one hydrogen ion. So two electrons from the food are removed and added to NAD+, and that makes NADH plus H+. For simplicity, it's usually just shown as NAD plus becoming NADH. And the food molecule is then oxidized. Then these electrons are used to provide energy, as well as ATP being formed. There's basically three stages to cellular respiration. The first is called glycolysis. The second is the citric acid cycle, and the third is oxidative phosphorylation. Next step is called glycolysis, and glycolysis takes place in the cytosol. And you don't need to memorize all the steps of glycolysis, but you need to know the net overall process, which I'm showing here. So in this process, glucose is being oxidized, and the product of that is called pyruvate. So glucose is being oxidized, so electrons are being taken from the glucose and added to NAD+, and that forms NADH. And overall, you get two NADH formed, and there's also a net production of two ATP molecules from two ADPs and two inorganic phosphates. And because glucose is 6 carbons, from each glucose you get 2 pyruvate, because the pyruvate is 3 carbons. So there you get two of those for each glucose. And then that's really all you need to know about glycolysis. And the pyruvate is then brought into the mitochondrion, and then it's further oxidized. So this is showing the next step. It's pyruvate oxidation, and in this step the pyruvate is further oxidized. So NAD plus extracts electrons from the pyruvate and becomes NADH. And also, there's removal of one carbon as CO2 gas. So you go from a 3-carbon pyruvate to a 2-carbon acetyl molecule. And there's also a coenzyme that's added to the acetyl called coenzyme A. The next step is called the citric acid cycle. Citric acid cycle is taking place in the mitochondrion, and it starts out with a 4-carbon compound called oxaloacetate, and to that is added the acetyl-CoA. So the acetyl group of the acetyl-CoA is added to the 4-carbon oxaloacetate. And so that the acetyl group's 2 carbons, so you're adding 2 carbons to 4, so you get a 6-carbon molecule called citric acid. The citric acid is then oxidized and a CO2 is removed. So NAD plus gains electrons, becoming NADH, and the CO2 is removed. That forms alpha-ketoglutarate. Since we removed a CO2, we removed 1 carbon, so now we're down to 5 carbons. Next, the alpha-ketoglutarate is further oxidized, forming more NADH, and another CO2 is removed. And now some ATP is formed. And the process of ATP formation here is called substrate-level phosphorylation. That means that the phosphate is coming from the intermediates in the cycle. And the next step is, well, you have succinate, it's 4-carbon compound, and once again that's oxidized further, forming another reduced cofactor called FADH2. And the result is fumarate, and then that's hydrated to form malate, and then the malate is further oxidized to form oxaloacetate again. And then the cycle repeats, and each time the cycle repeats, another acetyl-CoA is brought into the cycle. So the overall process produces a lot of reduced cofactor as you're oxidizing the intermediates. And there's also CO2 produced and some ATP. After the citric acid cycle, we have the third and last step of cellular respiration called oxidative phosphorylation. So all of the reduced cofactor that's made in the citric acid cycle then goes to the surface of the mitochondrion. So the citric acid cycle is taking place in the innermost part of the mitochondrion called the mitochondrial matrix. The reduced cofactor goes to the inner mitochondrial membrane. So recall that the mitochondrion has 2 membranes, an inner and outer membrane, and there's a space between them. So in the inner mitochondrial membrane are a series of proteins called the cytochromes. These cytochromes take the electrons from the reduced cofactors, NADH and FADH2, and they transfer these electrons to each other down a chain of proteins. And as those electrons get transferred, there's also quinones, which are called mobile electron carriers. As these electrons get transferred, the electron loses some energy, and the energy that's lost from the electrons is used to pump hydrogen ions from the matrix side of the inner mitochondrial membrane into the intermembrane space. So these cytochromes pump these hydrogen ions into the intermembrane space. And the electrons ultimately are added to oxygen. So oxygen is the terminal electron acceptor. It takes the electrons from the reduced cofactors and is reduced in the process. And so oxygen gets reduced and it becomes water. So this is where the oxygen and the water comes in. And all of these hydrogen ions that accumulate in the intermembrane space then flow back into the matrix of the mitochondrion through a channel called the ATP synthase. And you can think of it as the potential energy of these hydrogen ions being released to form ATP. So ADP and inorganic phosphate are combined, and the energy from the hydrogen ions is used to form ATP. And that process is called chemiosmosis. So this is a second way that ATP is formed. We saw the substrate-level phosphorylation. It was one way to form ATP in the citric acid cycle and also in glycolysis. And then the second way to form ATP is chemiosmosis. And chemiosmosis generates a large amount of ATP. So we had a small amount of ATP formed in the citric acid cycle and in glycolysis, but chemiosmosis generates a lot more. It generates about 15 to 20 times as much ATP. So this is where most of the ATP is formed, is in chemiosmosis. So we've seen how we can acquire energy in cellular respiration using oxygen as a terminal electron acceptor. But what about environments where there's no oxygen? Do cells always die when there's no oxygen? Well, not necessarily. In some cases, it's possible to get energy in an environment that has no oxygen. And that's through a process called fermentation. So in fermentation, you start out the same way with glycolysis. So here's our friend glycolysis, and we're starting with glucose, oxidizing it to form pyruvate, and that generates the reduced cofactor and some ATP. But then in fermentation, what happens is instead of oxidizing the pyruvate is reduced. So you're adding electrons to the pyruvate, and there's a couple of ways that's done. One is to directly reduce the pyruvate to lactate or lactic acid. So certain bacteria can do this, and also our muscles can do this when they're being worked very hard. So long distance runners and swimmers will eventually their muscles are working so hard that their muscles actually run out of oxygen. And when that happens, they can continue to get some energy by reducing pyruvate instead of oxidizing it. And there's a buildup of lactic acid in that situation. So notice that when pyruvate is reduced, that regenerates the NAD+, so that glycolysis can continue because the NAD+ is replenished and you can keep going. Another way to reduce pyruvate is to reduce it in two steps. One is to remove one of the carbons to form CO2 gas. And then the second step is the reduction. And the reduction in this case produces ethanol. So this is used by yeast to produce ethanol, and this is how all of our alcoholic beverages are produced, is by yeast doing glycolysis, and then the pyruvate is reduced with formation of CO2 gas and ethanol. Now you may wonder, what's in it for the yeast? Are they just producing ethanol for our enjoyment? Well, no. The yeast produces ethanol because the ethanol can kill their competitors in their environment. So the ethanol is toxic to many of the yeast's competing microbes, other fungi and bacteria. So in an environment that's rich in glucose, the yeast can use the ethanol. How carbohydrates can be used to form ATP, but it's also possible to use other kinds of foods besides glucose, besides carbohydrates, to generate ATP, and that's because the fats and the proteins can be broken down and then used as intermediates in these pathways that we've described. So for example, fats can be broken down into glycerol and fatty acids. The glycerol can become intermediates in glycolysis, and the fatty acids can be broken down to form acetyl groups to form acetyl-CoA. And proteins can be broken down into amino acids, and then the amino groups removed. And what remains of the amino acid after removing the amino group can become the citric acid cycle intermediates. And also some can form the acetyl groups and even pyruvate. So by this way, the other kinds of foods, fats and proteins, can enter these pathways to produce ATP. The reverse is also true. So if you add ATP, you can use intermediates from these metabolic pathways to form fats and proteins.